this is where we left off last time. So talking about why genetic mapping is not necessarily the way you want to go. So when genetics first started out, before the structure of DNA was even known, Watson and Crick didn't solve that until didn't publish that until 1953. People were already making the genetic maps that we talked about so far, and they are all based on recombination frequency. So the frequency of recombination between two genes. The higher the frequency of recombination means the more often you get a crossover and recombination event between them, and that happens more often for the most part as genes get farther apart. But as you can see to this here, due to undetected double crossover events, which you can't see between two genes, the actual map distance ends up being a lot higher than the recombination percent. So undetected recombination events will artificially lower the recombination rate between two genes because you can't see them. And it's even worse because each double crossover event would count, if you could see them, as two recombination events. All right? So we've talked a little bit about genetic maps, and I've sort of mentioned the physical map. So what is a physical map? The physical map is just like if I was to start here and then write pages and pages and pages of A's, T's, G's, and C's. It's just the complete base, the DNA base pair sequence of the entire chromosome. Right? So genetic maps, as I said, are based on recombination frequency between two genes. So here on one chromosome are genes A and B. They are 2 map units apart. So what does that mean as far as recombination frequency? 2%, exactly. So if you do big A, big B over little a, little b and cross it to a little b homozygous recessive tester, 2% of the time you will get either big A, little b or little a, big B, right? Same thing happens with genes C and D. Again, these are 2 different chromosomes. They're not homologous chromosomes. So the same thing, wow, genes A and B are two map units apart, and genes C and D are 2 map units apart. Everything is cool until you find out what the actual DNA base pair sequence is. Uh-oh. We're off by like 1 million base pairs. So in reality, when you look at the physical map based on the actual distance in base pairs between the 2 genes, gene A and B are 1.5 million base pairs apart. But genes C and D are 2.5 million base pairs, apart. But they both have a 2% recombination rate. So either the recombination rate here is higher than it ought to be, or here it's lower than it ought to be. And actually it's the former. So what happens is throughout the genome, I've lied to you again. Remember I said if you have one end of the chromosome and the other end of the chromosome, you can divide it into 100. There's like a 1% probability of recombination in any one of those spots. That's not true. Due to chromatin structure, so how the DNA is complexed with histones, which we'll talk about in chapter 11, and packaged, can lead to certain spots in the chromosome called recombination hotspots. These are areas where recombination happens a lot more frequently than it would if everything was fair, right? So even in genetics, I'm going to tell you life's not fair, right? And for these, this one is a recombination hotspot. So a recombination hotspot makes this distance, which is 1 million bases smaller, appear the same as this one. So an artificially high amount of recombination here due to the recombination hotspot, actually is making genes A and B look farther apart than they really are. In some parts of the chromosome, I don't have a picture for it, but there would also be recombination cold spots. So recombination hotspot makes recombination more frequently than it should be. Recombination cold spot makes recombination occur less frequently than it should be. And if you think about it, you can tell that a hotspot would make two genes look farther apart, because the larger the frequency of recombination, the larger the genetic map distance between them. So in all organisms that have a sequenced genome, the genetic map positions are still known. Like in our C. elegans genome, when I go to the database, the genetic map positions are known, but you also have physical map positions for everything, for every gene that has been what we call cloned, or where the actual bases for that gene have been identified. So some genes are not yet cloned. Many genes aren't. And we just know them by, okay, well, if gene A is here and gene B is there, then the new gene is going to be between them, right? And so we know that it's between this spot on the physical map and that spot. So genetic mapping that I do in my lab and that other geneticists do is largely to put new genes that you're trying to map between genes that you already have on the physical map. So and to look at a somewhat realistic estimate of the distance that that gene is between them. But nobody in C. elegans ever goes and like does the three, and even in fruit flies, because their genome is sequenced too, nobody goes and does 3-point crosses and figures out the, genetic mapping down to that scale. In fact, a lot of people in C. elegans, now that genomic sequencing has gotten so cheap, barely do any genetic mapping at all. So I'm doing this right now where I have some new mutants and I know what chromosome they're on. I'm not even sure where they're at on that chromosome, but I'm just sending the mutant off with some genetic crosses that is too much to explain here, but a few genetic crosses, send the DNA off to China to get it sequenced, because it's now I can get for under $200 the entire genome sequenced, and then you can compare the sequence of the DNA with the mutant to the sequence of a normal worm, and you can find your mutants that way. All right. One way to place genes on the physical map is by mapping relative to other genes on the physical map, like I just talked about. A second way is to use deletion mapping, or also called deficiency mapping. So a deficiency or a deletion is where a part of a chromosome is missing. So just look at this guy right here. All right, so just look at this guy right here. See that this chromosome is shorter than the other ones? It's shorter because it's got a deletion in it. The red box that's shown in the other chromosomes, that sequence is missing. So this little piece here is hooked right on there. So this whole piece in red is missing from that one. So we can use deficiencies or deletions to map new mutants and put them on the physical map, because we know where the ends of these deletions are. So we're going to use a complementation test, same as we did before, only instead of crossing our new mutant to a tester that has an allele that we already know about and seeing if it's another allele of that gene, here we're crossing a homozygote for our mutant to a phenotypically wild type organism that is heterozygous for a deletion. So this chromosome is completely wild type. So it says A+ here. Every gene in the red area here is going to be wild type or normal. It's just showing A plus because we're talking about the A gene over here. This one is, you could think of it as being mutant for every single gene that's within that deletion. So we're not talking about is this new mutant an allele of gene A or is it an allele of another gene? We're asking, is this mutant, is the mutant A somewhere within this red box or not, meaning somewhere else? So here we're showing gene A actually being within the deletion. We cross it here. 50% of the progeny are going to, so everybody gets the mutant chromosome, and 50% of the progeny get the wild type chromosome there. So half of these ought to be wild type no matter what happens. And then half of them get the mutant chromosome and then the deletion. In this case, because the deletion contains the gene, now you're having little a over, it's not even over little a, it's little a over nothing, right? Because that gene doesn't even exist. So that's going to look like the mutant phenotype. So if you do this cross and you get half of the progeny having the mutant phenotype, you know that mutant A, the gene for it, is somewhere within that red box. And the red box is just, you know, for each deletion strain where that is. Down here, look, gene A is still up there, but this is a different deletion strain where the deletion is down there. This one's shorter because this piece right here is put on there. In this case, same as before, everybody gets a mutant chromosome, this one, and then half get the completely wild type chromosome, their wild type, and then the other half get the deleted chromosome right here. But because A gene is not within the deleted part, you're still wild type because this one can complement, the deletion chromosome can complement your mutation. If you were to, in most of these cases where you're doing the cross like here and here, you have to cross your homozygous mutant into a heterozygote for the deletion. Because the strain with the deletion is missing so many genes, generally that chromosome, if this is homozygous, you're going to be dead. So generally you have special strains where you have this heterozygote maintained and you're doing that. Does that make sense? So on these, just like the other complementation test, this is sort of backwards. I'm showing you what happens if A is within your deletion and if A is not within your deletion. But you don't know that. The point of deletion mapping or any complementation test is that you don't know the answer and you're doing the crosses to figure it out. What this is you know what's going to happen if your gene's in the deletion, you know what's going to happen if your gene's not in the deletion, and then you see which of those situations your results match more closely. Make sense? So this, if your deletion is within the mutation, you should get half mutant and half wild type progeny, normal. Over here, if your deletion is not or if your mutation is not within the deletion, you should get 100% wild type or normal progeny. Questions? Are we good? It's just a complementation test again, but the chromosome that you're trying to get complementation to, you can think of, instead of being mutant for just one gene, it would be mutant for every single gene within this region. So here's an example of mutational or of deletion mapping that I did when I was trying to clone or figure out the identity of one of these sterile genes. So this is a physical map of C. elegans chromosome 5. So UNC42, SPE10, FUR4, and SMA1 are, well, not FUR4, but these three are genes that have already been cloned. So the numbers above them are the physical map positions. it's in number of base pairs. So this one's at 9.7 million and this is 11.9 million. So I had mapped my new mutant called SPE42 between UNC42 and this other one called SMA1. And so I knew it was somewhere in this area on the physical map, but there's 2 million bases in there. So what I did was I found a bunch of available deletions, and 3 of them actually impinged on this area. So let's look at this one first. So all the deletions here, this is the end of the deletion, and it goes from here all the way out here, and the dots just mean it goes out to the left of UNC42, and we don't care because I already know my genes to the right of that. So S-diff, or deficiency, because that's what they're called in C. elegans, is missing everything that I'm circling right here. So I did a deficiency mapping or deletion mapping cross. So as all of your mutants are in the lab that we're working with, if you're homozygous, you're sterile. You make oocytes instead of eggs. No eggs on the plate, no babies on the plate. If I did a cross to put SPE42 over S-diff35, those worms were fertile. That means that the SPE42 gene can't be anywhere in this area that's covered by S-diff35, because if it was, there would have been no SPE42 plus, and it would have been sterile. So that means I already knew the SPE42 was between UNC42 and SMA1. Now I know it's between here and here. Not quite half, but that's almost 1 million bases that I cut off just with one cross. Then I tried the same thing with, unfortunately, this area of the genome's not covered very well by deficiencies. Here's a tiny one here. Here's another one. Here's another one there. So all of these SPE42 over deficiencies ended up being fertile. So I never really found out from the deficiency mapping where it is. I found out where it isn't. But by knowing where it isn't, you can infer where it is. So I had my area to look at knocked down to here and here. Man, there's a lot of I'm going to go check and see if the door is shut right. I thought they were all gone, but we're getting a, maybe they're not out there for you, but on the screen and in my face, I'm getting a love bug celebration down here. They're going full on all around, flying around my head. All right, so that's helpful. Now I know it's in one of those 2 areas, and I went on and did more genetic mapping after that, but this made it easy. Just with those crosses, I can cut off big areas. Now if I had had a deficiency that, say, covered this whole area, and that was fertile as well, I'd be like, all right, because that means I know that my gene is just in this tiny area here, right? All right, we're not going to talk a lot about human genetics because human genetics requires a lot of very sophisticated, high-end statistics, way over what you learn in biostatistics even. And so beyond me, and to be a human geneticist, you just about have to be a statistician, or you have to have one on your staff, like a trained, at least undergraduate statistics major, if not a PhD level. But what we are going to show is how you can do associations of genes with areas in the genome without doing the statistics to see what's going on. So when you're working with human genetics, you can't do a bunch of crosses, ethics and all. And also, even if you could, humans would be like the world's most lousy model organism. Because all the stuff that I talked about way back in chapter 3 that makes a good model organism, like short generation time, no. Large number of progeny, no. All these things, easy to culture under standard conditions, no. you know, in big numbers. None of that stuff. We're terrible. So what happens is we have to look at pedigrees and make inferences from them. So what we've got here is in the filled in circles and squares are individuals who have an autosomal dominant trait for what's called Nail-Patella syndrome, where they have either missing or severely reduced fingernails and kneecaps. So there must be something about making fingernails and, oh, they're out with you too now. There must be something about making fingernails and kneecaps that's the same, so this, you know, this gene goes there. It was shown through pedigree analysis that the Nail-Patella syndrome gene, or locus, is very close to and therefore linked to the gene for the ABO blood groupings that we've talked about. So remember, if you are little I, that's blood type O, that's recessive. Big I is going to be either big IA for type A, big IB for type B, or big I, well, yeah, that's it. And you can be big IA on one chromosome and big IB on the other one for type AB. So what you do to look at the try to estimate the map distance between these is you look at a whole lot of family pedigrees. And you know that this gene should go with the Nail-Patella syndrome. So here you have a person with blood type A, person with blood type B, and in their progeny they have blood type O down here. There's one and there's one. So if you have an A father and a B mother and a child who's O, you know that each of the parents have a little I allele too. So now you know the genotype of these guys perfectly. So nobody with blood type A has Nail-Patella syndrome. Everybody without blood type A has Nail-Patella syndrome. So because this parent doesn't have it, you know it's not on either of her chromosomes. The Nail-Patella syndrome allele is going to be on this guy's chromosome, and it's the N right here, the capital N, because it's a dominant disease allele. So the N is going to go with people who are either O or B, because they're going to get one of these two genes, but they're not going to get A. And in this case, for whatever reason, and it's pretty amazing because there's 1, 2, 3, 4, 5, 6, 7, 8 kids in this level, nobody gets AB, really? So AB would also not have it. So you're saying everybody, so it's on this chromosome, so everybody who gets this chromosome is going to have Nail-Patella syndrome, and then you go down and look, and you can see where you get crossover events. So by looking at individuals, you can tell that this male has the Nail-Patella syndrome on his little I chromosome, and when you follow it down, there's a problem here because now you have this person over here is type B, not AB, so that means they have Nail-Patella syndrome right here, and so this person has type B and that guy down there. Type A in their progeny, which is what this is, the only way you get type A, because this person here is type A, you get that chromosome, and you have to get the Nail-Patella syndrome chromosome too, right? Otherwise you'd be AB. And so this guy, this male here has got Nail-Patella syndrome, but this one doesn't, meaning that there had to be a crossover event so that you start here, little I, crossover and recombine, little I goes with little n right there, and then you would expect possibly, depending on the gametes, so that means there's a gamete out there but it just didn't get fertilized, that is type B with N, right? Just in that event. Likewise here, this person is type B. They have Nail-Patella syndrome. This person has children with a type A that is normal. So that means anybody who's B having this one should not have Nail-Patella syndrome. And that is the case here. However, everybody with type O should have it, because to get type O, you've got to get both of those, just down here. And this one doesn't. So in this person, you had a crossover and recombination event where little I goes with little n, big B goes with big N, and therefore you get that. So this is a very small pedigree of just 2 generations. So human geneticists take pedigree after pedigree after pedigree, like hundreds or thousands of, with lots of pedigrees with lots of children in them, and by combining a lot of those numbers together, they can get an estimate for how close together these actually are. With the human genome being sequenced, as long as you get somewhat close, you can sequence the area of the genome around the linked gene, which in this case is blood type, and try to find a difference in DNA sequence between people who have Nail-Patella syndrome and people who don't. And in that way you can actually get the gene. This is fine, except unlike C. elegans worms, where we have the dumpies, the uncs, the rollers, blistered, all kinds of mutants that we can look at, humans don't have all these kinds of mutations that are handy for mapping against. So what we generally end up doing is we map against what are called SNPs, or single nucleotide polymorphisms. So the human genome has been sequenced, right? There's a standard human genomic sequence, but all of us have many, thousands of differences from that, because mutations happen over time and accumulate. And unless a mutation gives an advantage or a disadvantage, evolution doesn't act on it. About 45% of our genome does not code for protein or RNAs, or is the areas of the genome in which you have those promoter regions we talked about that controls when genes get turned on and off. So about 45% of our genome is just floating out there. It's composed of repetitive DNA sequence that's been put in there over time by rogue DNA elements called transposons that we don't have time to go into. But you get a mutation in any of those areas and it doesn't hurt anything, right? One mutation versus not. So everybody will have a bunch of differences. So a single base change is like, one person has the normal genome has an A and I have a T, or the normal genome has a G and you have a C at that spot, right? So somebody who has, you get a family who has genetic disease, and you know sort of where it is, or even if you don't, you can just sequence these individuals and find out what their polymorphism status is. So in each of these cases, A, B, C, D is just an area. Each chromosome has a bunch of SNPs on it. So, you're looking, you're like, okay, well that's 1, 2, and 4, right? So there's only four possibilities for SNPs, because you can have an A, a C, a G, or a T. In some cases, you see even more. Like this C has a 5 here. How can you have a 5th type? Another type of difference between the chromosomes, it occurs when you have repetitive sequence. So like CAG, 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 or something like that. And it's how many repeats do I have versus how many repeats do you have and how many repeats do you have. So there's a possibility to have any number of repeats. In any case, SNPs can be used, or other polymorphisms can be used, just like genes. You can do genetic mapping with them. Farther apart two SNPs are, the more likely you get recombination between them. The closer they are, the less recombination you get between them. And by comparing the recombination rate of a certain disease allele to a bunch of SNPs, you can find out which one it's closest to. And all of these single nucleotide polymorphisms are on the physical map. That's the coolest part. They are right there on the physical map, so you know exactly where, or pretty close to where your gene's going to be at. All right, so look at this case. So here we have a mutant allele, D minus, they're just saying, right? So D is farthest away from A, and then B is closer, and C is the closest thing, right? So if you were looking at the recombination frequency between the disease allele, or somebody with the disease, and these polymorphisms, you would find out that D recombines with polymorphism A lot faster than it does with B and C, right? So then you would be able to say, well, D is closer to C than to A and B. And likewise, E, F, G, H, I, you know, stuff on the right-hand side, if there's E, F, G, H, I, you would know that D is closest to C and E, so that you know exactly where to look for here. So if you get crossing over like this, then you're good. Now the reason why you get less recombination between C and D is because, look, if you want to get recombination between C and A, you can have recombination anywhere between here and here, right? There's a lot more area there between C and D. So again, recombination frequency is used to pinpoint where things are at. Good? All right. So now we are going to be moving on to chapter 8 of abnormalities at the chromosomal level. And here's where, eventually, where you will want to look back at your, come on, there we go. Look back at your pictures. That's actually a fly. Look back at your pictures. I wish I had a pew, 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 pew. Wouldn't that be cool? I guess I would melt holes in the screen, but I got one of those fly guns at home, and I'll tell you what, I was skeptical, but I'm like, I'll drop $35 on this thing. And it was, oh my God, it's so much fun. Sit out, it's like you fill it with table salt, and it's like a little shotgun, and you pump it, and it's like an air gun, and the fly sits on your hot dog and go, poof, and the fly goes, poof, and just flies away. It's so rewarding. And it's just with salt, so you can shoot them off your food and then eat it. It's fine. A little seasoning there too. So you're going to want to look back at your pictures your pictures that you took when we did the chromosomal abnormalities in the lab. And again, I'm sorry that this is the one part where what we're doing in the lab and what we're doing in class is kind of disjointed, but because the C. elegans lab lasts so many weeks, there's not really any other way to do it, and students have said that building those beads, the bead chromosomes, actually helps a lot with understanding what we're going to talk about here. All right. So we're going to talk about chromosomal mutations. These are gross or big changes. Yeah? Okay, so yeah, I'm sorry, I forgot to change that. So here's the deal. In the fourth edition, so I guess I have to go back and change it. So it used to, so chapter 8 in our book is the chromosomal abnormalities. Chapter 9 we're not doing because that's microbial genetics. Chapters 8 and 9 used to be switched. So I think it was between the 4th and the 5th edition, and I never went back and changed it in the syllabus. It's like they switched the order in the book. And I never do the microbial genetics chapter because you cover that in microbiology, and I have other stuff here that they don't talk about. So So big time changes, big pieces of chromosomes moving around. Unlike the single nucleotide polymorphisms, which there's just a single base change that I just talked about, and even like the repeat numbers where you have a CAG, 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 for example, and you have a bunch of those put together, and how many do you have? Those are pretty small compared to breaking off big pieces of chromosomes. So we're going to talk about the structure of chromosomes first, and then we're going to talk about ploidy issues with the number of chromosomes coming up. All right, so the first thing we're going to talk about is chromosome rearrangements. So we've talked about this a little bit, like with the deletions we just talked about, inversions, we talked about in lab, and we're going to go over again, translocations, and some more that we're going to cover. Then the second thing is we're going to talk about aneuploidy. Aneuploidy and polyploidy both relate to the change in the number of chromosomes that you have. Aneuploidy refers to individual chromosomes. The one everybody knows about is Down syndrome. If you have 3 chromosome 21s, this is you have Down syndrome to some degree or another, depending on what the rest of your genotype is. That's a change in number of just one of the chromosomes. All the rest you have 2. Polyploidy, on the other hand, is alterations in the number of complete chromosome sets. So polyploidy, for example, is a diploid organism that has 2 copies of every chromosome becoming a tetraploid organism that has 4 copies of every chromosome. So changes in all, you're going to have the same number of every chromosome, that's just going to be changed. So because we're humans and we're diploid, and most other animals are diploid, we're going to use this as the standard. So N is haploid. I just put the 1 there for ease of understanding. 2N is diploid, and anything above 2N, so triploid, tetraploid, hexaploid, octoploid, etc., those are all going to be polyploid. All right, so this will be a general introduction, and then we're going to get into the chromosome rearrangements today. So here is a normal chromosome. It's 2n equals 6. There's 3 different kinds, 1, 2, 3, and there's 2 homologous chromosomes of each. Chromosome rearrangements is just a change in the gross structure of a chromosome. So you can see here, this one goes A, B, C, D, E, and here you have a duplication, and don't worry, we'll get to exactly what that is, more about it later on. This chromosome has an extra CDE stuck in right there, so it's longer. Everybody else is the same. Aneuploidy, as I just said, is a change in the number for 1 or maybe more chromosomes. However, it's not going to be all of them. So here is a trisomy. You can see while the red and the green chromosomes are still diploid, are still what you would expect for a diploid organism, this blue chromosome is a trisomy. Right? And I'll come back to that in just a second. So here is polyploidy. This is a triploid. So everybody has 3. I'm going to cover it and say this again, but I want to say it now so you kind of get it in your head. Trisomy is when you have 3 of one chromosome. Triploid refers to the ploidy of the entire organism, so everything. This is not trisomy, and this is not triploid. Triploid refers to the whole genome. Trisomy or tetrasomy or monosomy or nullosomy or anything with somy refers to changes in just one chromosome. And I'll get to the numbering too, so don't worry about that. So let's talk about chromosome rearrangements first. So you can think of your word processor, I guess, when you except for these 2 at the bottom. Duplications is where you copy and paste. So your chromosome, compared to the normal chromosome, the mutated chromosome or rearranged chromosome is going to have all the same sequence but then some extra. Deletions, as we talked about, are like just highlighting some of the chromosome and hitting delete. It's gone. It's taken out of the chromosome, and just like when you delete something in a word processor, the rest of the text comes up to join it. Here, when you've taken a piece out of a chromosome, the rest of the end comes back and joins where the piece was taken out. Inversions is where a piece of a chromosome pops out, flips around, and goes back in. We've talked about that. And same thing here with a translocation. So with a translocation, we're talking about swapping of DNA, and this is the important part here, between non-homologous chromosomes. You get swapping of DNA all the time. That's what recombination is. But the full name for it is homologous recombination. Homology means similarity, right? Like the chromosomes are homologs. So homologous recombination is what happens between two homologous chromosomes, the non-sister chromatids. That is swapping the same parts of one chromosome for the same parts of another chromosome that is its homolog. Translocations, as we saw in the lab, are taking a part of one chromosome and putting it somewhere else in the genome where it doesn't go. And sometimes another part comes back and sometimes it doesn't. Don't worry, we'll get to it. Let's start with duplications. So here's your normal chromosome, and then at the bottom you have a duplication. In this case, EF has been duplicated and then stuck in either there or there, you can't tell. So you have two of the area that has E and F. We're using this just as an example. Of course, this chromosome would have more than 7 genes, right? When you get a duplication of a chromosome, in almost every case, you're going to have more than 2 genes in that. So the E and F, you could just think of as regions, right? So the E region and the F region are going to be there. We've talked about gene dosage a little bit when I talked about dosage compensation, right? Where we talked about managing the amount of expression from X chromosomes. Animals, and to a pretty similar extent plants, are very, very sensitive to gene dosage, especially during development. If you are a diploid organism, you need the equivalent of exactly 2 doses of every gene. There are exceptions. We talked about the immunoglobulin growth factor where your mom's gene is off and your dad's gene is on, right? But for everything else, you want to have exactly 2 doses. If you have a duplication here, you get improper gene dosage. So look at this organism. If you pretend these are the chromosomes in an organism, they have 2 of A, B, C, D, and G. That's fine. But then because there was a duplication in the E and the E and F genes or regions are going to have 1.5 times the dosage. That may not seem like much, but when you're talking about the gradients of signals that are required for determining which end of you is your head, which end of you is your feet or your tail if you had one, which side of your hand has the thumb on it and which side of your hand has the finger, which side of your arm is the side that's out, developing the outside of your body, which side is the side that's closest. Just little changes in signaling ratio can mess things up. And if you are homozygous for this, then you would have 2 times the gene dosage. So gene dosage is a big deal. Also, position effect. So we talked a little bit about promoters, the DNA sequence that doesn't code for anything, but will turn on or off a gene. If, for example, if you take this and duplicate it over there, when you duplicate this part, you could be taking just the part of, let's call this a gene again, the E gene that's for the protein, or that's for protein coding, but the promoter was not taken with it. But when you put it in over here after F in this part, now there's a promoter there that's driving E that wasn't the one that normally was doing it. And this promoter is on all the time. And E is not supposed to be expressed all the time. Now you're in trouble because you have way overexpression of E, and again, you know, the consequences are up to and including you die embryonically. Or so it shows E and F here, and then E and F are going to go somewhere in there. What if there's a gene right here, and that gene gets broken in half when the E and F regions go in there? Now that gene doesn't work, and so you would only have one good copy of the gene up here, and the gene here, half the gene's over there and half of it's over here. It's broken in half and doesn't work. Get it? All right. So different types of duplications. Tandem is what I've shown. You just take it and put it right after. Displaced is here. You put it on the other end. And reverse, you have it somewhere, but it's flipped around in the opposite orientation. The displaced and reversed are especially vulnerable to the position effects I talked about, because now this E and F here is way out beyond the G, and you could have not the control region it should have or extra control region. Yeah. Oh, I've got a plant in the audience today, because the next thing I was going to say is, see down at the bottom there, how do you tell the difference between a reverse duplication and an inversion? Because that's inverted, isn't it? It's flipped around the opposite orientation, just like the inversion. So look here. I'll let you figure it out. So we're talking about the E and F regions, right? Let's look at the normal chromosome up here. Just look at this one on the top. Now, if you had an inversion involving the E and F regions, what would it look like? It wouldn't look like this, would it? You would just take this part out here, the E and F, you would take it out, flip it around upside down and backwards, and put it back in. So the normal chromosome would have one copy each of E and F. The inverted chromosome would have one copy each of E and F, right? How many copies of E and F are there down here? That's the difference. It's a duplication because the same chromosome has two copies of that region, and they just happened to be inverted when they got put back in. Are we good? We're getting not enough questions. And see, we had a question, we took care of it, and I'll bet somebody else thought that too, and you just didn't want to ask. So you should. Because if nobody asks, then it doesn't get answered. All right. If you have a duplication or a deletion, you get problems when chromosomes pair, because these are big pieces of chromosome. Chromosomes line up on the metaphase plate at meiosis 1 by lining up homologous regions, A, B, C, D, E, F, and G, and we have this loop forming because there is an extra E and F part that is not in the normal chromosome on the top, so it sticks out. Now, when we talk about deletions, you're going to get a loop forming like this as well, but in this case, when you have a duplication, the chromosome with the loop has the duplication in it. When we get to deletions later, the chromosome with the loop is going to be the normal chromosome because the chromosome with the deletion is going to be missing some sequence. But we have to talk about some other stuff first here. So let's look at duplication. So here's everybody's favorite fruit fly, and one of the mutants that was found was called bar eye. So here's the normal eye here. This is the bar eye. It's just, you know, it's a line like that. If you count, and somebody has done this, the number of eye facets, the individual little mirrors in a wild type fly region, you would have 779. If you are heterozygous for bar mutation or duplication, now instead of 2 dosage of the bar region, you have 3. And just that 1.5 extra, 3 doses versus 2, gets cut in half, the number of eye facets, right? If you are homozygous for bar, that means you have twice the amount. That's even more severe. If you have 4 copies of bar instead of the 2 you're supposed to have, you go from 780 eye facets to 58. That's a more than 10-fold reduction. That's pretty serious. And if you have an extra one here, well, actually, this is the same thing. So if you have double bar and then a regular chromosome, these are within, you know, statistical analysis. They're not significantly different. And so 4 copies. So this, what's actually happening here in the fruit fly is that you have to have genes for eye formation. So, and PAX6, which is one of the genes, don't write this down, but just, you know, so you can see, wow, making eyes is really complicated and all, right? And how do all the animals figure out how to make an eye? Well, the PAX6 gene is involved in all eye formation just about that we know about, from the really primitive eyes on the little crabs that have the little eyes around the front of them, to eyes on spiders, eyes on insects that have facets, to our eyes, which is just, you know, one eyeball with one lens and all that stuff. So what you have here, though, is the bar, normal function of bar is actually to limit the field of the eye, right? You don't want an eye over the whole part of your face, right? It's got to be right where it's supposed to be. But if you have too much, then the signal to make an eye and the signal for no eye here is out of whack. And areas that should be eye, you can see around the edge there, there, and there, aren't eye anymore, because the no eye here diffuses in further at a higher concentration, and so it prevents eye formation where it should actually happen. Yes, maybe not anywhere, but it's one of the things where if you express this gene somewhere else, you will get eye development. Will it be a fully functional, beautiful eye that the fly can see out of? Probably not. But you can change all kind of structures in the fly. Like you get another cool mutation that that is often shown in textbooks is where, like, one of the antenna that they come out that they normally smell with gets changed into a leg. So you signal for a leg, and now you get an antenna developing. If you have a frog which develops, you know, hands sort of, you know, a thumbed, you know, it's not a real thumb, but, you know, it's sort of a one side versus the other side here. If you take and put the signal, retinoic acid, for making a thumb, you know, for doing that on the other side, then you can get a hand that would essentially have a thumb on both sides. It starts the process there. All right, so how do you get duplications? One way you can get them is you can get a duplication and a deletion from what's called unequal crossing over. Crossing over and recombination is homologous recombination. The sequences have to be the same, but what I just got done saying is that a lot of our genomes are full of repetitive DNA sequence. And so the recombination can be screwed up a little bit, where there could be DNA both here and here on either side of the bar that looks enough the same that you could get a recombination instead of having recombination here and here or here and here, you get recombination on the opposite sides, right? And then if you use your finger to follow the chromosomes, you get bar, bar, in there, and this one's going to be up to here and go up there, and you have no bar. So right there, you get a duplication and a deletion. So bar is what the duplication is called. Then, once you have two of them, once you have the duplication, then all the sequence in both red areas is fine, and it's easier to get double bar, which is 3 copies, by doing this. So you get double bar and go back to just 1 copy there, and that's what this guy is. But then this fly can pass on the double bar chromosome to its progeny. Right. I am sad to say I wasn't asked to look over. I was asked to look over a few chapters for the new book, but not this one. And I forgot to tell them, but this mistake is still in the book, right? So this is the X that I put in. This is the X the book has in there, right? So so this is the red opsin and green opsin genes on the X chromosome. This is how you get red-green color blindness. You lose one of them. But what they're showing down here is this chromosome has a red and 2 green opsin genes, and this one has none. That one would be red-green color blindness. Getting this is impossible if you use the recombination X that's shown there. Look, red, red, green. So you'd have a red, red, green, and this one is coming up here, no red and green. So you'd have one red, red, green, and one with green only, and you got the opposite. So they are wrong. So cross this off and put an X there right in your book, so if you sell it back, somebody else gets it and they're not here, they don't get screwed up. Look, red, green, green. That's that one. Nothing, red, nothing, right? Right there. So either the artist screwed this up or they screwed up where the X goes. All right. So in a deletion, part of the chromosome is missing. Here, E and F part is missing. And now we get back to just what I was talking about previously. But if you line up 2 homologous chromosomes and one of them has an insertion, you'll get a little bulge. If you line up 2 chromosomes and one of them has a deletion, you'll get a little bulge. But in this case, the chromosome with the bulge here, and this is what it would sort of like look like in real life, is the normal chromosome. It's bulging out because the deleted chromosome is missing the E and F genes, and so it goes A, B, C, D, G. So if I showed you a picture that looked like this but didn't have any colors on or anything, they were just gray lines all the way over, and I said, what's this bulge? There's 2 possibilities. Either this chromosome with the bulge has an insertion and the bulge, or a duplication, and the bulge is the duplication, or this chromosome is normal and the other chromosome has the deletion and the bulge is the normal part. So you can't tell the difference. But you would know it's either one or the other of those. So having deletions can lead to a phenomenon called pseudodominance. So here's a wild type C. elegans worm. Dumpy over dumpy is dumpy over dumpy is a dumpy worm, short and fat. But just like what we did with the deletion mapping we talked about earlier, if you put a gene or a chromosome that has a dumpy mutation in cis to another, or in trans to another chromosome that is missing the entire region, it has a deletion for where the gene is located, so the blue region here is gone there. These worms would be dumpy too because you have a dumpy allele here and you have no allele there. So this is called pseudodominance. The normally recessive phenotype is expressed because there's no wild type gene there. So any mutation for any gene that is in this blue region here would look mutant if you put it over that chromosome that has the deletion in it. Yeah? So pseudodominance, term you have to know. Another term you have to know is haploinsufficiency. So haploinsufficiency is sort of incomplete dominance type phenomenon, but you would get a separate phenotype for it. So there is a gene in the Drosophila called Notch. It's a very important signaling receptor, like a gene for a signaling receptor. It is so important and required for development that if you are a notch over notch homozygote, you are dead. You die embryonically and you never become a fly. So these are pictures of the end of a fly wing, and the reason why it's called Notch, and the only reason it was able to be discovered as a mutation, is that as a heterozygote, you're missing one notch, so you only get one dose instead of 2 of the notch protein. It's enough to make you develop, like you don't die as a ball of cells right after fertilization, but you get this little notching in the end of the wing there. And this is just like the yellow mutation in the mice. So fruit fly people were originally in a sort of a conundrum because they're like, what's going on here? Because they found the notch mutation. And notch mutation is going to be dominant over wild type, so as long as you have one notch, you're fine, but you could never get a notch homozygote. If you cross 2 notch flies, you could never get a true breeding notch fly. All the babies would produce 2/3 notch and 1/3 wild type because notch over notch is dead. Exact same thing. So if you have a 4-square Punnett square, 1 would be plus over plus, 2 squares would be plus over notch, that's 3, and then the 4th square would be notch over notch and it would be dead. So you would have 2 out of 3 and 1 out of 3 here instead. And so this haploinsufficiency is a case where just having one wild type dose of a certain gene product is not enough to get you to the wild type phenotype. You can see this a little bit in some of your worms, but you're probably not looking for it. So some of the dumpies and some of the unks, and I don't think dumpy 11 is too bad at this, and I know none of your unks are, but in some cases, like there's another dumpy that's called dumpy 5 on chromosome 1. I don't use that with you because if you are plus over plus, you're wild type, dumpy over dumpy, you are a very small, really short dumpy. And if you are dumpy over plus, you are supposedly wild type, but if you look closely, you can tell that you're just a little bit shorter and fatter than normal, and that's going to be, again, that's going to be sort of this haploinsufficiency or, incomplete dominance there. Okay, so we'll see you for lab if you have it today. If you don't, I will see you on Thursday. Please make sure that you have the lab stuff looked over, I'm going to go over it. But if you are seeing it for the first time when I explain it to you today, you will have a hard time understanding what's going on. Now that we have discussed, now that we have talked about linkage, it should be a lot easier to understand what's going on, and that's another reason why I waited to explain it to you until now. All right, see you in lab.